Hello, everyone. Our second speaker is Allison Cruikshank. She is a PhD student from Duke University. Her talk is Sex Differences in Liver Cell Metabolism and Their Consequences. Allison, please go ahead.
Thank you so much for the introduction. I'm excited to give this talk today. So yes, I'm a fourth year PhD student at Duke University working with Dr. Michael Reed in sex differences and their consequences and oxidative stress management. Okay, so this is a schematic diagram of our model, so liver cell metabolism. So you can think of this as a typical liver cell. So we have the cytosol in the blood compartments. We don't include any other organelles like the mitochondria or peroxisomes for this first model. But I'll go into a model later that has the mitochondria. But yes, we see that we have the folate cycle here, the methionine cycle, and then we have the glutathione pathway down here in purple. But what you see here, so the rectangles are represent substrates, and the ovals represent enzymes that drive the reaction. So this reaction right here, denoted by the black arrow here, converts DHF to THF, and the enzyme that does that is DHFR. So we have a bunch of biochemical reactions in this figure. All the black lines are biochemical reactions or transport from the blood, like you see here with cysteine. And then we also have these long red arrows. So these are allosteric interactions in this pathway. So you can see this substrate over here, SAM, actually inhibits this enzyme way over here in the folate cycle, MTHFR. So this inhibition is denoted by this line with that horizontal line. And then we also have, for example, betaine over here stimulates this enzyme, CBS. And these stimulations are denoted by these red lines with the arrowheads at the end. So we had these biochemical reactions and then these allosteric reactions as well. So the first part of this pathway is one carbon metabolism, like I said. And for this talk, the molecule I want you all to focus on for this one carbon metabolism pathway is homocysteine. So homocysteine is basically the molecule that connects one carbon metabolism to the glutathione pathway down here. And homocysteine is important because it's a major biomarker for cardiovascular disease. And actually, women of childbearing years have lower levels of homocysteine than men in postmenopausal women. And previous mathematical models of our group have actually helped to explain this phenomenon. Um, so you might be wondering, well, how do we take into account the sex differences in this model? Well, all the enzymes that are highlighted yellow are known to be upregulated or downregulated by estrogen. I'll be using the term estradiol to denote estrogen in this talk. But yes, all the yellow enzymes are affected by estradiol. So we're able to put those in based on the literature values and see how it affects different substrates. So the main purpose of my talk is this pathway. So glutathione is GSH down here. It's made up of three amino acids, glutamate, cysteine, and glycine. So knowing this, the synthesis kind of comes easily. You first, homocysteine is converted to cystathionine, which then is converted to cysteine. Then you add a glutamate, and finally that last amino acid, glycine, is added to make glutathione. And then glutathione can be oxidized, so it takes in this oxidative stress up here. This is constant in our model. It makes the oxidized form, which is glutathione disulfide. So that's just it's just basically two glutathione molecules fused together by a disulfide bond. And then this oxidized form can then be reduced by glutathione reductase to again just make glutathione. So glutathione is important because it's a main antioxidant in the body and conjugates and eliminates a variety of reactive oxygen species and toxicants. And actually, in the liver, glutathione removes heavy metals and toxicants from food. So you could kind of think of it as it has it's an evolutionary mechanism to in the fight of humans against plants in the history. But interesting, Lee, it take oxidative stress is connected to this glutathione pathway. This oxidative stress is constant in this first model. I'll go later on in the talk, I'll just introduce a new model that actually takes into account oxidative stress and define it more. But for this first set of results, you can just remember it as it's bad at high levels, basically. It contributes to the risk for heart disease and cancer. So that's all I'll say about it, say for it right now. But importantly, in this glutathione pathway, you can see that two enzymes are highlighted yellow. So estradiol, E2 is the abbreviation for estradiol that I'll use throughout this talk. It actually stimulates glutathione cysteine ligase and glutathione peroxidase. So those enzymes are actually upregulated by about 80% in females relative to males. So we're able to put that in our mathematical model. So our goal with this first model is to investigate the mechanisms underlying sex differences in glutathione metabolism. And actually, so all the rectangles are actually, so those are substrates, like I said earlier, and we have a ODE for each of those. So it ends up being a system of like 30 ODEs. The enzyme velocities act as rates in those ODEs. So all the ODEs follow mass balance. So um, the rate, it's just a rate in minus the rate out, basically, of each of these substrates. And all the enzymatic rates take a Hill function or Michaelis-Menten form. So they're highly not, it's a system of 30 nonlinear ODEs. Okay, so that's basically all I'm going to say about the math part of it, but I wanted to get into the results basically of this mathematical model now. So our first result is looking at estradiol's influence on glutathione. So you see here a glutathione steady state curve. So we just increase estradiol and calculate the glutathione steady state at different estradiol concentrations. So that's the y-axis right here. And all the concentrations are in micromolar. So the average estradiol concentration in males is 0.09, and that's relatively constant throughout their lifetime. So the glutathione, we're able to plug that estradiol concentration into our model and get that the glutathione concentration in males is 5,000 micromolar. However, for the premenopausal female, the estradiol changes throughout the menstrual cycle. So actually, the max for the average menstrual cycle, the max estradiol concentration is 1.2 nanomolar, and the minimum is 0.15 nanomolar. So that's the significance of this estradiol range I chose for this graph. But if you take the average estradiol along the menstrual cycle for premenopausal women, you get that the average estradiol concentration is 0.4368 nanomolar for women. So that puts them at this point right here. And you can see that in the premenopausal female, the glutathione concentration is significantly higher than that of males. And that's actually a result that's seen in the clinic and in humans and in mice and rats as well. So we're able to replicate that. But interestingly, is what you don't see as much in the literature is how glutathione reacts to estradiol. So like at different concentrations. And we see here that it actually behaves non-monotonically. So it doesn't monotonically increase, which you might think from looking at the diagram I showed earlier. Um, and actually, interestingly, at very high estradiol concentration, the glutathione concentration for premenopausal women actually falls below that of males. So high estradiol concentrations can maybe be bad in a way. Um, but we can use our mathematical model to explain why we see this non-monotonic behavior. And I'll explain why using what we call this glutathione regulatory motif. So it all takes into account the fundamental idea that at steady state, which this curve is basically the steady state at different estradiol concentrations. At steady state, the flux in has to equal the flux out. So the flux in the glutathione has to equal the flux out. Um, so actually, when estradiol first starts to increase on this side, um, the synthesis of glutathione is going to increase, driving, so the flux in is going to increase, driving glutathione up. However, once you get to a certain point, glutathione is going to start to inhibit glutathione synthase, GS, and glutamate cysteine ligase, GCL. So by inhibiting those, it inhibits its own synthesis. So the flux in is going to decrease. Now, this enzyme glutathione peroxidase is still increasing at the same rate that estradiol is increasing. So in order for the flux in to equal the flux out, glutathione is going to have to decrease to balance those. So that's why we see this decrease. So we're able to explain that concept with the mathematical model and the mechanism behind these results. Um, Now this shows us the basic effect that estradiol has on our on glutathione in our model, and that's the substrate we're most interested in right now. However, during the menstrual cycle in premenopausal women, estradiol doesn't monotonically increase. So this isn't what the glutathione curve would exactly um, look like during the menstrual cycle because estradiol during the menstrual cycle actually follows this curve. So you can see the maximum estradiol concentration is about 1.2 nanomolar. The minimum is about 0.15 nanomolar. And we take an average 28-day menstrual cycle for this model. So we wanted to calculate what glutathione does during the menstrual cycle. And I actually show you two results that we get. So let me explain the difference. So this first result on the left is what we call the curve of steady states. So that's basically calculating the steady state of our model. So calculating the glutathione steady state concentration at each 1/10 of a day. So at each 1/10 of a day, we plug the corresponding estradiol concentration in, and then calculate the steady state. And that gives us this glutathione curve over the menstrual cycle. It actually varies from about 4,000 micromolar to 9,000 micromolar. Now, this is kind of like the naive calculation, which you might first go to calculate what glutathione does during the menstrual cycle. However, this is what you actually get if you plug this periodic solution into this periodic estradiol curve into our model. So if you plug this in and run the model for I think at least four menstrual cycles, you'll get that glutathione will always stabilize to this periodic solution. And actually, you can see that it varies. It's actually much more stable than what you might realize. So, but I mean, what you might think with this naive approximation. So, glutathione actually only varies from about 55 micromolar to 8,000 micromolar. So it's much more stable than what you might think, and you could think of this as maybe an evolutionary um, adaptation. And because it might not be good for glutathione to vary that much throughout the menstrual cycle.
I
Um,
think I could you just say again what the difference was in the right plot?
yeah, yeah. So for the right plot, we don't calculate the steady state. We don't calculate the steady state at each estradiol concentration. We basically we let estradiol vary and then the solution of glutathione actually stabilizes through this periodic solution. So you could think of it, so what's causing this difference is that at every time we change estradiol, glutathione is trying to reach the steady state that corresponds with that. However, before it can reach that steady state, estradiol changes again. So you can kind of think of this as this solution as maybe like an average of this steady state curve. Does that make sense?
Yeah, yeah, thanks.
Yeah, and this is a interesting mathematical question, I will say. I did spend a bit of time like looking at whether we could prove this is like a global attractor. However, you kind of have to start small with this because we have like I said, we have a system of 30 nonlinear ODEs. So um, it might not be so easy with that hard of a system. So I was like starting off with like a two-state system and but didn't get any significant results, but it's still an interesting question, I will say. Um, but yes, this is a pretty cool result that we're able to show how stable glutathione is during the menstrual cycle. Um, so these are kind of the intrinsic results of our model. And now I want to go to an application of our model. So this application was introduced to us from this doctor at Duke in the medical school, Dr. Ayako Suzuki. So she came to us with the problem of homocysteine levels during hormone supplementation in pre and postmenopausal women. So if you remember homocysteine from that first schematic diagram I showed you, it's the molecule that connects one carbon metabolism to the glutathione pathway. And it was a key biomarker in cardiovascular disease. So what she saw when doing some analysis of the NHANES data is that serum homocysteine levels increased upon estradiol supplementation in premenopausal women. However, in postmenopausal women, serum homocysteine levels decrease after estradiol supplementation. So her question really was, why do we see this differentially effective estradiol on homocysteine levels in premenopausal and postmenopausal women? You might think since it's interacting with women, we're looking at pools of women, they might behave the same. However, it's key that one pool is premenopausal and one pool is postmenopausal. So the estradiol concentrations are different before estradiol supplementation. Um, so she came to us with this question. She knew that we had that the mathematical model that had the glutathione pathway and homocysteine, and she wanted to see if our mathematical model could help to explain this phenomenon, since she was quite puzzled by it. So the question is, how does glutathione influence homocysteine levels? So this is, we use the same exact model as I was talking about earlier. Um, However, this is just a zoomed in look at the things that are important in the explanation of my answer to this question. So I there are two new things in this in this mathematical model that answers this question. We first include the effect of progesterone. So progesterone, the abbreviation is P4. That actually decreases glutathione peroxidase activity. However, the decrease is very small. So the estradiol stimulation actually dominates that physiologically in liver cells. So it doesn't create that much of an effect in our model, but we thought to include it because she had other results I won't show today that our model helps shine a light on. So the other main thing that we added is that glutathione stimulates this enzyme CBS. So we didn't include it in the previous models because we didn't know that this mechanism existed until we started. When she came to us with this question, we knew that we had that glutathione non-monotone curve with estradiol, so we thought that has to be there has to be like some connection there. So we are scouring literature looking for glutathione's effects on different enzymes like upper in the one carbon metabolism pathway that might affect homocysteine levels, and we found that glutathione actually stimulates CBS. So if you imagine if glutathione increases, there'll be more stimulation on CBS, meaning so there's going to be a greater activity of CBS, meaning that homocysteine is going to have to decrease in order to balance the flux into and out of homocysteine. Then when glutathione decreases, there's going to be less stimulation on CBS, so homocysteine is going to have to increase for the flux in to equal the flux out at steady state. So it's kind of like that intuitive explanation that's going to explain these results coming up. So this shows some of the results on the differential effect of estrogen supplementation on homocysteine levels. So this is in premenopausal women. So I show the glutathione concentrations, the steady state concentrations, and the homocysteine steady state concentrations. Um, so for premenopausal women, we take the average estradiol concentration to be 0.4368 nanomolar. That's just like I said, the average estradiol concentration during the menstrual cycle in premenopausal women. So before estradiol supplementation, glutathione's at the top of this curve here, corresponding to that average estradiol concentration. However, upon estradiol supplementation, glutathione levels fall. This decrease in glutathione causes an increase in homocysteine. And that's because of the reason I was discussing earlier in that previous slide. It comes from glutathione. Since glutathione decreases, there's going to be less stimulation on that CBS enzyme, so homocysteine levels are going to increase. Now, in postmenopausal women, sorry, these dots should line up. I don't, I don't know what happened. So in postmenopausal women, the average estradiol concentration, so in postmenopausal women, they don't have the menstrual cycle, so the average estradiol concentration is the same as that of males, 0.09 nanomolar. So before estradiol supplementation, their glutathione level is the same as that of the males in our model, um, and in the clinic as well. But upon estradiol supplementation, glutathione increases then because it's on this side of the this side of this glutathione curve. So this increase in glutathione causes a decrease in homocysteine. So homocysteine, that this explains why homocysteine levels decrease in postmenopausal women. So, and all of these results are able to provide a mechanism, an explanation for why we see this differential effect of estrogen supplementation on homocysteine levels in premenopausal and postmenopausal women. And it was a very cool project to work on, I will say, because it was my first time working with uh, a real life medical doctor in helping to explain some clinical results. And it was very exciting working with her. So um, very proud of this work. And it's currently under review right now at PLAS One. So hopefully the paper's out soon. Okay, so lastly, with this end of this talk, I want to talk a little bit about this current work I'm working on. So sexual dimorphisms and liver oxidative stress. So oxidative stress, if you remember from that big diagram I showed on that first slide, it interacts with glutathione to make the oxidized version of glutathione disulfide. So in the previous model, oxidative stress was just constant. We just took it to be 0.01 micromolar because that's what it usually is in liver cells. But you might be asking, what is oxidative stress and why am I even why do I care what it is? Why do I care about making a mathematical model of it? So oxidative stress is an imbalance between oxidants and antioxidants. Glutathione's an antioxidant. And this imbalance leads to accumulation of reactive oxygen species. And excessive reactive oxygen species cause cellular dysfunction and disease. So they're important signaling molecules at physiological levels, but as always, everything in balance. So excessive levels cause cellular dysfunction and disease. And actually, this is a diagram from this paper down here. So this is a mitochondria. And a cell. And you can see that oxidative stress is implicated in cardiovascular diseases, neurological disorders, metabolic diseases, and diseases associated with aging. So it's an area of ongoing research, and there's a lot of research going on that's showing that oxidative stress is more and more important in a lot of these diseases that we see. And actually, importantly, women have lower levels of oxidative stress. So that's known in the clinic, in humans, and in other mammals like mice and rats. They're able to measure these levels of oxidative stress and see that women have lower levels. And actually, I thought I would just throw this out there because it's very interesting that I found when I'm starting this work about 2 years ago, is that a lot of scientists out there have been accumulating a lot of evidence that they think that this is the main reason, or not a main reason. This is a contributing factor as to why women live longer than men on average. So they think that oxidative stress plays a big role there. So that would be that's that makes it a very interesting area of research just in that itself. Okay, so with that, I want to show a mathematical model of this oxidative stress. So I created a mathematical model for hydrogen peroxide, which is H2O2. So it's a major reactive oxygen species in the body. So we think of that as the indicator of oxidative stress in our model. Um, so you can see in this model, we have the cytosol, the blood, but we also had the mitochondria. So hydrogen peroxide is mostly produced in the mitochondria, and that's because in the mitochondria we have the electron transport chain. So in the electron transport chain, if you remember from your high school biology class, um, electrons may fall off of it. These stray electrons can create oxygen radicals, which are then converted into hydrogen peroxide. So a lot of hydrogen peroxide production occurs in the mitochondria, and then a smaller amount on the cytosol. So those are these arrows here. And importantly, in this model, we do not consider the folate cycle or the methionine cycle. We actually start our ODEs at homocysteine now. We don't take into account one carbon or anything further up than homocysteine because we wanted to focus on hydrogen peroxide, and there's not a lot of evidence right now that hydrogen peroxide affects those other one-carbon metabolism, or one-carbon metabolism affects allosterically affects enzymes in this pathway. But if there is, we can always just add it. So what I want to show here is that we have three scavenging mechanisms for hydrogen peroxide. So three ways to get rid of it. So like I said, high levels of these reactive oxygen species are bad. So we don't want high levels of hydrogen peroxide. So we have to have these scavengers to get rid of it. So it's actually present at like 0.01 in the cells, actually, in liver cells. So we have the glutathione pathway here, which is what I've been modeling before, but we also have these other two pathways. The thioredoxin pathway acts very similarly to the glutathione pathway in that we have a reduced thioredoxin being oxidized to oxidized thioredoxin, and then it's reduced back to the reduced form. And then this catalase enzyme is actually very different than the glutathione and thioredoxin systems. This catalase enzyme actually just directly just removes hydrogen peroxide to make water. So it's very efficient in that way. And it's very important at high levels of hydrogen peroxide. And then we also have homocysteine inhibition on some of these scavenging mechanisms like glutathione peroxidase and catalase. Importantly, these inhibitions do not go into effect until like very high levels of homocysteine. So we really only included this in this model because we wanted to model a disease, but I won't have time to talk about that today. But if you are interested in that, I'm happy to talk to you more about that later. But yeah, our goal was to investigate and uncover mechanisms for sex differences in oxidative stress management. So we wanted to analyze the effect of estradiol and metabolites at steady state and during the menstrual cycle. And then this is a disease I was talking about. So I wanted to study CBS deficiency and how sex plays a role in biomarker levels. So CBS deficiency basically is a deficiency in this enzyme CBS, which causes high levels of homocysteine. So you can already see that high levels of homocysteine will reduce the scavenging mechanisms, the activity of these scavenging mechanisms, leading to accumulation of oxidative stress. And that is indeed what's seen in CBS deficiency. But unfortunately, I won't be able to, I'm going to skip over the results of this model because I don't have time. I included them here because I didn't know if I would have time, but I don't think I have time. But future work actually is I want to analyze oxidative stress in pregnancy. So in pregnancy, placental tissues produce more reactive oxygen species with increased metabolism. In preeclampsia, which is a disorder in pregnancy, reactive oxygen species are in abundance throughout the body. So these placenta cells produce these reactive oxygen species, and then these extracellular vesicles carry these reactive oxygen species to organs like the kidney, liver, and brain. So that's the liver is where my work would come into play with looking at how oxidative stress takes form there in preeclampsia. Um, and then also I would want to look at the effect of oral contraceptives on oxidative stress metabolism as well. But this is all these two things are more further down the line, but I will say the results I skipped over just before this slide, hopefully it'll be out in a paper soonish. We'll see. But yeah, with that, I want to thank my collaborators, my advisor, Dr. Michael Reed, and our biological collaborator, Dr. Frederick Nyhout. And thank you all for listening, and I'm happy to take any questions.